In this video, we are going to discuss the final macromolecule that is important for life, which are proteins. Now, I am sure you have heard the term protein before, and some have a sense of how complicated they can be when talking about diet and nutrition. We are going to keep it simple and talk about the basics of what proteins are, starting with their structure. Now, when we say the term protein, we are referencing a large polymer structure with many subunits attached to each other. But before we can grasp that, we need to understand the structure of those individual subunits, which for proteins are called amino acids. The structure of an amino acid is very specific and contains a few parts that you need to memorize and be able to draw for the IB exam. Let's start with the middle of the structure, which contains one carbon atom, which we call the alpha carbon. Stemming from the alpha carbon are 4 single bonds that attach to other components that complete the amino acid. The orientation of the amino acid is usually drawn with an amine group branching to the left of the alpha carbon, which is a nitrogen also bound to 2 other hydrogen atoms, and a carboxyl group branching to the right of the alpha carbon, which is another carbon that has a double bond with an oxygen and a single bond with a hydroxyl group, which is an oxygen with another hydrogen bound to it. The next 2 parts stemming from that central carbon are a single hydrogen atom and a variable group that we represent with the letter R. This R is just a placeholder for a larger structure that is attached at this point. But when we are drawing a general amino acid, we do not know what structure is attached. So we put an R there to represent where that structure would be if it were attached. When comparing macromolecules, I always look for the straight NCC chain to help me determine if I am looking at a protein. This is a telltale sign that you are looking at an amino acid, which again are the building blocks of larger protein structures. We discussed condensation reactions in the last video talking about carbohydrates and lipids, and the same process is used for proteins. Proteins are built by attaching individual amino acids together to form long chains. So if we take 2 amino acids as seen here, you will notice that the carboxyl group of one amino acid can bind with the amine group of the other. The OH on the carboxyl group and the H from the amine group will condense into water, which forms a new bond between the carbon and nitrogen, linking the amino acids together. This bond between amino acids is called a peptide bond. And with 2 amino acids linked together, we just created a dipeptide. If we continue to link them and make a longer chain, we call it a polypeptide, which can then fold into its final form that we call a protein. Amino acids are very important structures that support living systems. Because of this reason, it is important that our cells have a mix of all of these important amino acids. We talked about the structure of an amino acid earlier, which includes that variable R group. When we look at the different amino acids, which differ in that R group, you need to know that there are 20 different possible versions of that variable group, which means that there are 20 different amino acids that our body can use to build proteins. Of the 20 different amino acids, some of them can be synthesized within our body and others can't. But remember that we need them all. So how do we get the ones that our body can't make? The answer is our diet. The food that we eat contains amino acids that the body can use to build proteins. We call amino acids that the body cannot create essential amino acids because it is essential to have them in our diet because we have no other source of obtaining them. The others that we can synthesize are called non-essential, which means that even if we do not get them in our food, our body can still create them from other amino acids. When considering different diets out there, it is important to take into account the types and amounts of amino acids you are getting. For example, consuming animal products like eggs, meat, and milk contain levels of amino acids that are similar to what the human body needs. If you are a vegetarian and do not eat meat or consume these products, you will possibly not have the correct balance of essential amino acids taken in from your diet. Taking supplemental pills or planning your meals accordingly with the proper amino acid concentrations in mind can ensure that your body is getting what it needs. Amino acids put together create a polypeptide, and when that polypeptide folds into its final form, we can call it a protein. The human body creates and contains thousands and thousands of different proteins that perform different functions. But how is all of this diversity possible? Well, remember that there are 20 different amino acids based on the different R groups. So if the cell is building a protein chain, it has 20 options of amino acids to use. We pair that with the fact that amino acids can be linked together in any order, and that polypeptide molecule size can vary. It creates a scenario where a massive amount of options are possible. If we do some math like we did when talking about the possible combinations that DNA can make, we can see that for proteins, the diversity is great. If I have just 2 amino acids with 20 options for each, I would have 20 raised to the second power, which is 400 different options just between those two. With a chain of 5 amino acids, there would be over 3 million different combinations of proteins you could make. You will encounter different protein structures throughout the course, but for the sake of naming some examples, we can look at insulin and amylase. Both of these molecules are proteins, but differ in amino acid length and order. Insulin is made up of 2 different protein chains that are 30 and 21 amino acids long, which is a relatively small protein. Amylase, on the other hand, is much larger, with a single chain of 496 amino acids. We'll talk about these proteins and their functions in more detail in later videos, but for now, just know that proteins can be extremely different in size and order of amino acids. Proteins perform many different functions around the body and within cells, and their ability to work properly is greatly dependent on their 3-dimensional structure. When amino acid chains are made, the final step before the protein is functional is for parts of the chain, including the R groups, to interact with each other and fold into a final 3-dimensional form. Assuming this structure is maintained, the protein should be able to perform its designated function. But the reality is, protein 3-dimensional structure is subject to change based on environmental factors. If something does impact the original structure of the protein and alters it, the protein will likely no longer work properly. We call this phenomenon denaturation, when an external factor negatively alters the shape of a protein. Both temperature and pH levels can denature proteins. If temperature increases beyond a certain point, it can denature proteins by breaking the bonds that hold them together. Similarly, if pH of the environment is altered too much in either direction, high or low, ionic bonds between R groups can be broken, resulting in denaturation. While this is the case for the extremes, keep in mind that different proteins have different tolerance levels for temperature and pH. Some single-celled organisms live in places under extreme heat, like near geothermal vents, and they make specific proteins that can function under those conditions. We see the same thing with pH by looking in your stomach, which is acidic. The protein enzymes found in the stomach that help break down food thrive in the lower pH condition as they are designed for it.